Well, welcome into this week's Degrees of Science. This week we've got some out-of-this-world research being done to help preserve Earth's biodiversity on the moon. So today we're talking with Dr. Mary Hagedorn. She's a senior research scientist from the Smithsonian Institute. So Dr. Hagedorn, I guess right off the top would be, so y'all's plan is to take cryogenically frozen material, biological material, and put it on the moon. I guess the first question is, why is it important to have cryogenically frozen biological material in the first place?
So we were actually inspired by the Svalbard Global Seed Vault, which put aside very important agricultural material as seeds in this very deep mine in Norway, in Svalbard. And mine is deep and it's very cold. It's the perfect temperature for storing seeds. And they don't need any people there to maintain it. They just put the seeds there and then they basically close the door. It's to act as a backup in case some of our very critical agriculture is destroyed by some, catastrophe here on Earth. And so we wanted something similar for animals and plants that cannot be dried as seeds. And there's no place on Earth that's cold enough because we have to maintain cryopreserved animal cells at minus 200 degrees Celsius. And seeds are kept at about minus 20 degrees Celsius. So we thought about going into a satellite, but there's a lot of radiation there. And then we started looking at the moon and the possibility of putting it in the south pole of the moon, which is very cold all the time and liquid nitrogen temperatures. And we could then bury it under 2 meters of moon soil, and it would protect it in terms of radiation.
Is this something that's kind of just a pie in the sky kind of thing, or is this something that we could actually see moving ahead anytime soon?
That's a really good question. We hope that it will. We are slated now for the very first step of this process, which is to actually measure the radiation that the cells might incur if they were to leave the Earth and go to the South Pole of the Moon. We have a spot on the Griffin 1 mission, which may be leaving next September 2025 for the South Pole of the Moon.
Is there a particular kind of cell that you're preserving that will be taken up there?
When you think of animals and all types of animals, we don't actually know how to cryopreserve many of them, certainly the sperm of a lot of them. Humans, we do a really good job with in terms of eggs and embryos and sperm and just a whole host of things, but we don't have that sort of menu of cells that we can do for other wildlife species. So we talked about it and we decided we would use the skin cells. And there's these cells within the skin called fibroblasts. And the fibroblast cells can be turned into very potent stem cells. They are very potent stem cells. And they can be turned into sperm and eggs in the future. And so we know how to cryopreserve fibroblast cells from almost every organism because they're uniform. And so that was the reason why we chose those cells, and they'll make a great library of cells. We think of them as kind of the book of life that we'll be putting on the moon.
So y'all are testing out with a reef fish called a starry goby. Is there a particular reason y'all are working with that fish?
It was a bit of convenience. I live and work in Hawaii, and I work on coral reefs. My colleagues from the Smithsonian were with me in Hawaii at the time, and we decided we would just take some fin material from the starry goby and learn to cryopreserve the fins and put it in the Smithsonian. But also, fish are very important. They're probably one of the groups that we do send, into the biorepository. But also, it's not an endangered fish. It's easy to get. So you want to work with something that's going to be not exactly a model, but something that's really easy to do so that you can have lots of material to test over time. And so it met a lot of criteria that we were going for. We wouldn't want to use something like, a cheetah or something like that where it'd be really difficult to get cells repeatedly.
So you're talking about taking something to space, and there's a lot of different things we got to deal with in space, the radiation, microgravity. Is there ways other than just kind of putting on the moon and testing it that we can kind of figure out if this would work up there?
Yes, that's another really good question. So if in fact we do get the funds to build our instrument to go into space, we will get radiation readings. We're going to have this tiny little box that is, think of it as like a little cryo freezer that will go up there, and it's going to have a little cryo engine on it. and it's the box is to try and mitigate radiation for the cells. If we do a good job, we'll have radiation sensors inside and outside the box that can tell us what level the cells will get on the way to the moon. And then we can test it on Earth. We can take that level of radiation and take the cryopreserved cells and expose them to that level of radiation, and then find out, are they damaged? Can they grow? Can we, just a whole host of battery of tests that we can do on those cells once we've exposed them to the radiation to know how they will do in space. And obviously, if we don't do a good job with the box, we will fix it and make it better.
So, cryogenically freezing, there's a lot of work that goes into that. So if you get it cryogenically frozen here and you send it to the moon, can it just sit up there forever just being at that temperature?
When you cryopreserve something, you basically halt it. You halt all cellular processes pretty much. And so it's in suspended animation, and it can stay that way for a day, a year, 10 years, even, theoretically even 1,000 years. And so you've just halted all biological processes. So yeah, they can stay there as long as there isn't a lot of radiation getting to them. They should be the same as when we put them, in that state.
When you start thinking of working up there with these cells that are preserved up there, you're talking about everything being that cold. How hard will it be for people to work around it, equipment to get this set up there?
Yeah, it's going to be a huge challenge, and I started wrapping my mind around that as well. I mean, think of Alaska, and in the real dead of winter, and your car battery goes dead with just a few hours, and you have to keep it plugged in. Or if you had to dig a hole outside your house in the middle of the winter in Alaska or something like that, it's colder than that, And so I think the challenges are going to be severe. But I also know that our engineers all our engineers on this planet are extraordinary. And the materials and the brains and the creativity that they are putting towards, working and living on the moon is really amazing. So I have full belief that, the robotics and things that they will use to do some of these tasks will be amazing.
Did you ever think, with a background like you have in biology and cryobiology, that you would be working with people in space and all this kind of stuff and designing something that would potentially go to the moon?
Yes, only because I applied to NASA as an astronaut when I left graduate school. So I'm a wannabe astronaut. So it's always, and I think they're the people who are on our team are just fascinated by space, And so it's a level of creativity and fun that we are having that isn't in our ordinary job.
That's really cool. So you've got a lot of organisms to pick from on Earth. When you're kind of getting this process going, are there specific areas or types of animals or organisms that you're going to want to first put up there?
I'd like to think of it as a living sort of museum. And to some extent, we are going to pick functional groups. Like, we want to keep ecosystems functioning. And I think the one that's easiest for people to understand is that probably most of the pollinators on Earth will be in our first group because they're so important to agriculture and life on Earth. There's many other things that you can think of that are really important, microbes that make oxygen. There's just a whole host of things. And so fortunately, I won't be the person who's choosing. We'll have probably an international team that will make choices, but it'll probably be about groups. And hopefully over time, we will hopefully have tens of years to do this. Maybe we will be able to put all species that are on Earth there, but that's a tall order. So we will start out with some really important groups like fish and pollinators and perhaps some endangered species, culturally important groups. There's a whole host that, like coral reefs and sequoias and things that just change our environment and may be important for terraformation of outer planets like Mars or wherever.
Very interesting, I think, exciting research that you are doing. Dr. Hagedorn, I appreciate you taking the time to talk with me.
Complete pleasure. Thank you for inviting me.